अस्सलामु अलैकुम स्टूडेंट्स, वेलकम टू माय चैनल इन्फॉर्मेटिव कन्वर्सेशन। स्टूडेंट्स, आज इस वीडियो में मैं आपके साथ शेयर करने वाली हूँ MLT मेडिकल लैब टेक्नीशियन मोस्ट इंपॉर्टेंट वाइवा क्वेश्चंस विद आंसर्स। तो वीडियो को कंप्लीट वॉच कीजिएगा एंड शेयर विद इट योर फ्रेंड्स। Question number 1. What is the purpose of quality control in clinical chemistry? To ensure the accuracy and precision of laboratory test results. Question number 2. What is an internal quality control? Procedures performed within the laboratory to monitor test performance. Question number 3. What is external quality control? Participation in proficiency testing programs to compare results with other laboratories. What are control samples? Non-samples tested alongside patient samples to monitor assay performance. How often should quality control be performed? Daily or with each batch of test? What is the role of a Levy-Jennings chart? To plot quality control data and monitor trends and shifts. What is Wester's rule? A set of criteria used to determine if a test run is in control. What is the purpose of proficiency testing? To assess the accuracy and reliability of laboratory results compared to other labs. What is a reference range? The normal range of values for a specific test in a healthy population. Question number 10. What action should be taken if quality control results are out of range? Investigate the cause, correct any issues, and repeat the test. Standards. What are standards in clinical chemistry? Substances with known concentrations used to calibrate instruments. Why are standards important in clinical chemistry? They ensure the accuracy and reliability of test results. What is the primary standard? A highly pure substance used to prepare a solution of known concentration. What is the secondary standard? A solution whose concentration is determined by comparison with the primary standard. What is the difference between a standard and a control? A standard calibrates instruments, a control check test accuracy. What is the purpose of calibration curve? To establish the relationship between instrument response and analyte concentration. How often should standards be used to calibrate instruments? Regularly according to the manufacturer's guidelines. What is a traceable standards? A standard that can be linked to a reference material of known accuracy. What is the role of the National Institute of Standards and Technology? To provide reference materials and standards for calibration. What is the purpose of a matrix matched strand? To match the simple matrix, ensuring more accurate results. Differentiate between quality control and standards. What is the main purpose of quality control? To monitor and ensure the reliability of test results. What is the main purpose of standards? To calibrate instruments and establish accurate measurement scales. How often are quality controls used? Daily or with each batch of test. How often are standards used? Periodically according to calibration schedule. What do quality controls monitor? The performance and accuracy of test procedures. What do standards establish? The relationship between instrument response and analyte concentration. What is the composition of a control sample? Similar to patient samples with known concentration of analytes. What is the composition of a standard? A pure substance with a precisely known concentration. Can quality control be used for calibration? No, they are used to verify accuracy, not calibrate. Can standards be used to monitor test performance? Indirectly, by ensuring the calibration is accurate. Topic: buffer solutions. What is buffer solution? A solution that resists change in pH upon the addition of an acid or base. Why are buffer solutions important in clinical chemistry? They maintain a stable pH for accurate biochemical reactions. What are the main components of a buffer solution? A weak acid and its conjugate base, or a weak base and its conjugate acid. What is the Henderson-Hasselbalch equation used for? To calculate the pH of buffer solution. What is the purpose of a pH buffer in enzymases? To maintain optimal pH for enzyme activity. How do you prepare a buffer solution? By mixing a weak acid with its conjugate base in a specific ratio. What is the role of bicarbonate in blood buffering? It maintains the pH of blood within the narrow range necessary for physiological functions. What is the pH range of a phosphate buffer? Approximately 6.8 to 7.4. Why are buffers used in electrophoresis? To maintain the pH and conductivity of the medium. What is the buffering capacity? The ability of a buffered solution to resist changes in pH. एंड स्टूडेंट्स हमारे पास इसके बाद कुछ टॉपिक्स रिमेनिंग रह जाएंगे जो कि हम नेक्स्ट वीडियो में वाइवा के सेकंड पार्ट में इन तमाम क्वेश्चंस को डिस्कस करेंगे.